Assalamu alaikum. Dear viewers, greetings. In this video, we are going to see about the bacterium Chlamydia psittaci. General characteristics of Chlamydia psittaci. It is gram-negative bacteria and shape is rod shape, non-motile in nature. The size of Chlamydia psittaci is 0.5 micrometer and belongs to the family of Chlamydiaceae. Capsule and endospores are absent. Respiration is aerobic. Optimum temperature is 56 degrees Celsius and pH is 7. Habitat common in variety of birds including wild birds, pet birds. Identified by J. Ritter in 1879. Pathogenesis of Chlamydia psittaci. Chlamydia psittaci infects humans when they or inhale dust containing dry droppings or respiratory secretions from infected birds. Transmission can occur through bird bites or beak to mouth contacts. Human to human transmissions of Chlamydia psittaci is rare. The incubation period of Chlamydia psittaci is 5 to 14 days. Virulence factor of Chlamydia psittaci, outer membrane protein, type 3 secretion systems, intracellular replication and inclusions, antigenic variation. First one is outer membrane protein. Chlamydia psittaci has outer membrane protein that help it to interact with and enter host cell during infection. Second one is type 3 secretion systems inject protein into host cell and manipulating their functions for the bacterium survival and replication. And third one is intracellular replications and inclusions. Inside host cell, Chlamydia psittaci forms specific inclusions where it replicates, creating a safe space that help it to avoid the immune system. Fourth one is antigenic variations. Chlamydia can change its surface antigen through antigenic variations, allowing it to hide from the host immune response and stay in the body for a long time. Pathogenesis of Chlamydia psittaci. Psittacosis is transmitted to humans through inhalations of respiratory secretions or bird droppings, particularly birds like parrots. The bacteria Chlamydia psittaci enters host cell, typically in the respiratory and digestive tract, using the process like endocytosis. Once inside the cell, the bacteria forms characteristic inclusions within the cytoplasms. These inclusions serve as specialized compartments for bacterial replications and protections. Chlamydia psittaci goes through a two-phase development cycle, switching between infectious elementary bodies and replicative reticulate bodies within these compartments. Inside the inclusions, Chlamydia psittaci actively multiplies using the host cell machinery for reproductions. The bacterium can move from one host to another, often avoiding exposure to immune factors outside the cell. This way of living inside cell helps the infection persist. Chlamydia psittaci also trigger an inflammatory response in the host, causing the release of cytokines and chemokines. This response attracts immune cells to the infected area, leading to tissue damage. In severe cases, the infection may spread throughout the body, affecting various organs and causing complications such as pneumonia, hepatitis, or neurological issues. Clinical disease caused by Chlamydia psittaci. Psittacosis. Psittacosis caused by Chlamydia psittaci. It is a respiratory disease in humans transmitted from infected birds like parrots and other birds like pigeons. Ducks can also carry the bacterium. The symptoms of psittacosis include respiratory symptoms. Psittacosis typically shows flu-like symptoms such as fever, chills, headache, muscle aches, and fatigue. Cough. Patient may experience a persistent cough, often accompanied by difficulty in breathing. Atypical pneumonia. Chlamydia psittaci is a cause of atypical pneumonia, which differs in symptoms from typical bacterial pneumonia. Systemic complications, whole body issues. Besides respiratory symptoms, severe Chlamydia psittaci infections can lead to systemic issues like hepatitis and myocarditis. Hepatitis. The infections may affect the liver, causing symptoms like jaundice and liver dysfunctions. Myocarditis. In rare cases, inflammations of the heart muscle has been linked to Chlamydia psittaci infections. Neurological complications. Although uncommon, Chlamydia psittaci has been associated with neurological issues including meningitis and encephalitis. Ocular manifestations. Eye problem. Chlamydia psittaci infections can rarely affect the eyes, leading to symptoms such as conjunctivitis or other eye-related manifestations. Laboratory diagnosis of Chlamydia psittaci, serological test, complement fixation test, microimmunofluorescence test. Complement fixation test measure antibodies level in a patient's serum. A rise in titer can indicate recent infection. Complement fixation test may be less sensitive or specific compared to other methods. Microimmunofluorescence test uses fluorescent label antibodies to detect specific antibodies in the blood. It identifies the Chlamydia species causing the infection and is often more sensitive than complement fixation test. Nucleic acid amplification test, polymerase chain reaction, a molecular biology technique amplifying and detecting Chlamydia psittaci DNA, highly sensitive and specific. It identifies the pathogen in clinical samples like respiratory secretions or tissues. Culture. Chlamydia psittaci is challenging to culture due to its intracellular nature and specific growth requirements. Specialized cell culture systems like HeLa cell may be used, but it less common due to technical complexity and time. Direct fluorescent antibody staining. Staining involving fluorescent label antibodies in patient sample is a relatively rapid method. It's useful for identifying chlamydia antigen directly in respiratory secretions. Chlamydia psittaci is an obligatory intracellular pathogens not growing on standard culture media. Traditional tests like colony morphology and biochemical tests are not applicable. Consulting with healthcare professionals and clinical lab is crucial to determine the best diagnostic approach based on clinical presentations and available methods. Treatment and preventive measure. Antibiotic theory. Infection caused by Chlamydia psittaci should be treated with an effective antibiotics like tetracycline, macrolides like azithromycin, erythromycin, chloramphenicol, rifampin. Preventive measure. Wash hand with soap and water after handling or cleaning up after birds. Purchase pet birds from reputable sources. Isolate infected birds. Consult a vet for treatment and follow guidance on reintroducing them to other bird safety. Avoid overcrowding birds in cages or enclosures. Clean birds cages daily including changing food and water bowls. Wet surface before cleaning to minimize dust. Set up bird cages to prevent the spread of infection through contaminated food. Features dropping. Use solid barriers between adjacent cages. Avoid stocking them. That's all about the bacterium Chlamydia psittaci. Thank you for watching this video.